So we have Dr. Bone, who is a staff scientist at SLAC in Stanford. Her research areas of interest include biogeochemistry, geochemistry, and water treatment processes and separation science, which we get to hear about today. She seeks to tackle difficult multi-scale molecular to landscape scale environmental problems by leveraging cutting-edge synchrotron-based imaging and spectroscopy and scattering techniques to investigate processes that improve environmental health and resilience in the face of climate change. And that's one of the reasons why Sharon's at SLAC, is so she can use all of the tools there that are available to her. She's leading a research effort that's funded by us here at NAWI, focusing on kinetic modeling and experimental valuation. So Sharon, please feel free to take over and share your presentation. Oh, Sharon,
sorry, I forgot to unmute myself for a second. Thanks for that introduction. Yeah, so thanks everyone for attending today, and I'm really happy to share this work that we've been doing, funded by NAWI to predict kinetics of scale formation. So before I get started, I wanted to give an overview of the team. So we have a pretty big team working on this topic. At SLAC, I wanted to specifically emphasize the work of Eleanor Spielman's-Son, pictured here, and Charlene Gompier, pictured here, who were the postdocs who really led this effort. So another key collaborator on our team is OLI, and they have been developing the kinetic models that are really at the heart of this project. And this work is led by Gareth Das and Andre Anderko. And then at NETL and University of Pittsburgh, respectively, Nick Seifert and Radosław Fidich have been leading the work on this project to validate our kinetic models using laboratory-based systems. And Ritesh Pawar was a graduate student, now graduated, who did a lot of the experimental work for this. So our objective as part of this, that's funded by this NAWI project, is to accurately predict the type and rates of scale formation across all treatment steps of desalination, from feed solution to brine concentrate, through a fundamental investigation of mineral nucleation rates. But before I get into the kinetic model, I want to take a big step back and get a global overview of why this problem is important. So this figure comes from the recent IPCC report, and it shows the global distribution of water scarcity. So areas that are brown are locations around the world that experience water scarcity 12 months of the year. And if you look at the US, you can see that really a large portion of the US it is water scarce for more than half the year. And so this really emphasizes the fact that in the United States, in order to sustainably supply all our needs, all our water needs, from agricultural to municipal to industrial, we're going to have to work on the development of new technologies. So membrane-based technologies are going to continue to be an important part of addressing this water scarcity problem. Reverse osmosis is, of course, the most common membrane-based water treatment process used for desalination. And there you use high pressures to push water through a water permeable membrane, leaving the solutes on one side and collecting the water on the other side. There are other pressure-driven processes like nanofiltration, and then membranes are used in numerous other water treatment applications that are electrochemical, bioreactors as well. In all these instances where membranes are in use, a key challenge is scaling and fouling of those membranes. So this occurs when solutes and colloids accumulate on the membrane. If they precipitate out in a mineral form, that is called scaling specifically. For reverse osmosis, this increases hydraulic resistance and osmotic pressure. It decreases salt rejection. For all membrane processes, scaling and fouling necessitate cleaning and replacement. A recent estimate of for RO specifically estimates that fouling and scaling contribute 10 to 20% of operating costs. So I'm sure many of you who have joined this webinar are familiar with the problem of scaling and fouling. It's been well known as a problem for decades. It's been really well studied, and yet it still remains very difficult to predict fouling and scaling. And I would argue that that's because it's a complex process. So if we think about just scaling or fouling on a reverse osmosis membrane surface, for instance, as pictured in my little cartoon here, we have flow across the membrane surface that generates a concentration gradient so that the concentration of solutes or colloids at the membrane surface is different from that in the bulk. So you need a mass transport model in order to predict those concentrations. This has to be done for real feedwaters where you have a really complex mixture of solutes and colloids present. And at the membrane surface, these can form a variety of materials, both crystalline materials that are really well characterized thermodynamically, and then sorbates, adsorbates, absorbates, and amorphous phases, which may not be characterized well thermodynamically. And finally, these systems are not often at equilibrium because you have continual flow continuously bringing material to the surface. And so kinetics are going to be a very important aspect of determining which phases are present. So before I get into the sort of meat of this presentation, I want to give you an example of how I got started in this space. So I'm actually a geochemist by training. I work a lot on heterogeneous environmental samples like soils and sediments. And so to me, sort of the natural way to start investigating this process is just to simply ask the question, what is there? If we look at all the various materials that are present on a membrane surface, what forms? And then from that, we back out, okay, well, what models are we aiming to produce? So this image here is a synchrotron-based micro X-ray fluorescence image of calcium, silicon, and potassium on the surface of a membrane that was harvested from a pilot system operated by Orange County. And they were kind enough to provide us with a couple different modules. And so we harvested and looked at the composition of their surface. And what I really just want to point out here is that we did a comprehensive study of the calcium speciation across this surface. And what we find is representatives of all of these different materials that I just spoke about, right? So we observe metastable phases like amorphous calcium carbonate. We also find sorbates that are not well characterized thermodynamically. So calcium adsorbed to organic matter, adsorbed to silica. That was ubiquitous across the membrane. And then finally, we did have a bunch of crystalline materials as well, but we had difficulty rationalizing the presence of these minerals based on the simple mass transport models that we were able to access. So this brings us back to the necessity of building these fundamental models for mass transport, thermodynamics, and kinetics, and to do this in systems, or to do this ultimately for systems that are complex. So in this presentation, I'm going to focus on methods that we can use to look at kinetics for crystalline or non-crystalline systems, and also chemical structure for crystal or non-crystalline systems in a very fundamental way, such that we can start to build these models that then can be applied in complex systems. So our NAWI project is broken down into two different parts. And so I'm going to separate those parts out in this talk. The first part includes the development of a kinetic model for scale formation. And our approach involves, characterization of the rates of particle formation, development of a kinetic model to describe those rates in terms of the concentration of the the ions in solution, and then finally validation of that model in laboratory scale systems. The second part of this project is aimed more at characterizing sort of materials that are non-crystalline, that aren't well described thermodynamically, and trying to model those systems. So for the kinetic model, we're specifically looking at calcium, barium, and strontium sulfates, and that's the work that I'm going to present today. We focus on those minerals because it's an important class of scale-forming minerals in numerous feedwaters, from municipal water to groundwater. And then in the second part, where we are doing this more phase and stability characterization, then I'll be focusing on silica-based materials, and in particular metal silicates. So let's get started with the kinetic system. So again, our goal is to predict the rates of scale formation for mineral nucleation at variable saturation state and salinity. And those two parameters are really important because we need to cover a broad range in order to be able to predict the kinetics in both the feedwaters entering an RO system and in brine concentration processes that are being developed to treat the brine concentrate that is the waste product of reverse osmosis. And so I'm actually going to start by talking about the kinetic framework that we use in this project. And so this kinetic framework has been developed by OLI, and it is derived from classical nucleation theory, and it's a sort of an empirical usage of this theory. Basically, it relates the induction time for particle formation to the saturation index of that mineral for a given set of experimental conditions. So induction time here is an operationally defined term. It's the time required for particles to first be detected in solution. And the saturation index is defined as the logarithm of the ion activity product over the solubility product. So it expresses a ratio of how much mineral forming ions you have in solution to what the concentration of those ions should be at equilibrium. So it's an expression of the thermodynamic driving force. And basically, the sort of important thing to know about this is that these two parameters are really linked by a surface energy parameter that's not easy to calculate or to measure. And so we're deriving it empirically through a relationship by fitting the relationship of induction time to saturation index. OLI has also incorporated into their model a term that accounts for the delay in particle formation caused by adding an antiscalant or an inhibitor compound. And the way this compound acts is by amending that surface energy parameter. And the way it amends the surface energy parameter is developed by parameterizing a Langmuir type adsorption isotherm for the different inhibitors. And the plot I'm showing you here shows you this relationship between induction time and saturation index that can be derived in the case of the inhibitor NTMP or nitrilotrimethyl phosphonate using previously published data. So at SLAC, what we have been focusing on is enhancing OLI's kinetic model by using a technique called small angle X-ray scattering to determine induction times for the class of minerals that we're examining. A lot of the prior data that's available on particle formation has been done using turbidity, which is a visible light scattering method. SACS is an X-ray scattering method, and X-rays are, their wavelength is on the same order of magnitude size as that of the critical nuclei when they first form, and so it is a sensitive probe of when those particles first form. We do these SACS experiments at a synchrotron where we have a very intense X-ray beam that we shine on the sample, and we detect the X-rays that are scattered by that sample, which is a flowing suspension. And we detect the X-rays that are scattered at small angles on a detector that is placed 2.5 meters downstream of the sample. So if you've come across using X-ray diffraction, just XRD, during your time working with samples, this method is physically fundamentally the same. So in X-ray diffraction, you're using scattered X-rays to characterize the distance between crystal or planes in a crystal lattice, and that gives you information on the mineralogy. In this case, the scattered X-rays that we're looking at, because since we're looking at very low or very small angles, we're sensitive to distances that are on the order of 1 nanometer to about a few hundred nanometers, and so those sizes are on this order of size of a particle, and so that's what we're detecting. So we collect one of these SACS patterns every 3.5 seconds, and we starting at about 30 seconds, and we just do this continually and watch the particles grow over time, which can be seen by the increase in intensity of that SACS pattern. The image on the sort of the bottom left hand of the slide shows our actual experimental setup, which is very simple. We have a vial, we add calcium chloride or strontium or barium chloride, we add sodium sulfate under constant mixing, and then we have a peristaltic pump which takes that solution and brings it into a quartz capillary tube, and that quartz capillary tube works really well for enabling us to get that X-ray signal out of the sample. All that said, at the end of these experiments, what we have are a series of patterns that look like this plot on the left, where as you go from blue to yellow, you're increasing in time. Now, what you can see right away is that, especially on the left-hand side of the plot at what we call low scattering angles, you're really getting an increase in the slope, right? And that's indicative of particle formation, particularly large particle formation. We can actually quantify the number of particles present in this solution or in the suspension using what's called the invariant. It's basically just the integrated area underneath the SACS curve. And then we can plot that invariant as a function of time, and that really is plotting for us the number of particles in solution as a function of time. So that is directly the rate of particle formation. So in this plot here, I'm showing you that invariant in black versus, and that's overlaid with the concentration of calcium 2+ in solution, which is measured using an ion selective electrode. What I'd like you to see from this image is that we're monitoring the invariant only at the first stages of particle formation, right? So the calcium 2+ concentration doesn't start to come into equilibrium with gypsum until, well after 80 minutes on this plot, whereas, the number of particles in solution grows extremely rapidly between, say, 0 and 20 minutes. So we're really probing the beginning of gypsum formation, far from equilibrium. Another way to look at this system is using cryo-electron microscopy. So these images were collected on gypsum particles by flash freezing suspensions of gypsum at different time points. So 4 minutes after mixing, 6 minutes after mixing, and 15 minutes after mixing. And what we can see by characterizing the distribution of particle sizes, so particle widths and lengths at these different time points, is that you have particles that are growing anisotropically might be the word. So they're growing, the width grows very slowly, going from 60 nanometers to 77 nanometers between 4 and 15 minutes. That's the median width. But the length jumps from 280 nanometers at 6 minutes all the way up to well over a micron at 15 minutes. So the particles were sort of longer than the field of view in these systems. And so what that tells us is that in this first 15 minutes of reaction, these particles are not only nucleating, but they're growing, and the mechanism of growth is changing, leading to rapid growth along one axis between 6 and 15 minutes. And we think this is likely an assembly mechanism. So now let's compare what we see from the cryo-EM to what we see from the SLACs. On the left-hand side, the bottom left, I'm showing you the invariant and calcium 2+ plot that you saw already. And then on the right-hand side of the plot, I'm zooming in just on that time scale that we've probed using cryo-electron microscopy. So the orange, green, and blue bars show you, morphologically what's going on for the invariant at those time points. And I think the important point to make is that the invariant, even at these short timescales, is expressing not only particle formation, but it's expressing particle growth, and particle growth by a mechanism that's non-classical. So we have a conceptual model for gypsum nucleation and growth based on our work as well as previously published work on the early stages of calcium sulfate nucleation or formation. And we think what's going on is that calcium and sulfate ions form ion clusters. These are prenucleation clusters that are amorphous. Those condense into these nanoparticulate needles, and those then assemble into rods. When we're doing our kinetic model, we're looking only at the step where we go from these prenucleation clusters to these intermediate nanoneedles. So in particular, what we're doing, we look, all of the invariants show a characteristic shape where they have zero slope versus time for some amount of time, and then they have a positive slope. And that we think is the particles going from prenucleation clusters to particles. So what we do is we take the logarithm of that invariant, we fit, and then we fit a line with zero slope and a line intersecting it, and where that intersection occurs, we define as the induction time. Using this method, we can then apply this to calcium, strontium, and barium. We can do this from 0 gram per liter added sodium chloride all the way up to 200 gram per liter added sodium chloride. So sodium chloride falls out of solution at about 200, little above 250 grams per liter. So we're getting close to that saturation point. And we've also done this in the presence of different concentrations of the anti-scalant NTMP. So this data is then fed to OLI, and they refine the parameters for their kinetic model based on this SACS data. And so I'm showing you here OLI's model specifically for gypsum. So our next goal, now that we have, we've developed all of this kinetic data, or we've produced all of this kinetic data, we've developed the model for it, and then we have to ask the question, okay, these models were developed under homogeneous conditions. We just have a calcium and sulfate mixing in a beaker. How representative is that going to be when we have a system where we have pressure and flow and the presence of membrane surfaces where which can also potentially impact scaling. So NETL and University of Pittsburgh have been running different types of laboratory scale reactors. At NETL, they've been running osmotically assisted reverse osmosis. The University of Pittsburgh been running nanofiltration experiments. For both of these, they run experiments where they monitor the flux decline and for calcium sulfate, the conductivity decline over time. And then they run parallel experiments under the same solution conditions where they harvest the membrane at different times before and after our predicted induction time from OLI. And so I'm going to show you just a little bit of data from the OARO system. So these, before I show you the actual experimental data, these are plots of the OLI's predicted induction time as a function of saturation index and salinity for specific conditions that we've tested in the OARO reactors. So a couple things to notice: the relationship between induction time and saturation index is highly nonlinear, and there's we also observe a minor relationship between induction time and sodium chloride concentration. So after we harvest the membranes, after these different time points, we characterize the concentration of calcium distributed across the membrane surface. So the top set of images on the slide shows you the concentration of calcium as a function of time at 8, 21, and 50 minutes. And then in the plot below, I'm showing you the same data, but presented in a different way as the average and standard deviation of the calcium concentration distribution over time. And what we see is that at times close to the induction time of 5 minutes, we have substantial amounts of calcium sulfate built up on the membrane. We can compare this experiment, which was done at a saturation index of 0.47. This is, again, calcium sulfate and 25 grams per liter sodium chloride to 1. That was done at a slightly lower saturation index, but higher salinity. Under the conditions shown on the right-hand plot, the induction time comes in at about 7 minutes, and we were able to harvest the membranes before the 7-minute time point, and we can see that we have really substantial amounts of calcium sulfate built up on the membrane in this case. So there is calcium observed on the membrane close to and even a little bit before the induction time. Nonetheless, taking this data together with conductivity the declining conductivity, the kinetic model adequately predicts scale formation for this system. So this was for the calcium sulfate, these homogeneous conditions were a partial success for the laboratory-based reactors. This was not the case for silica. So that silica data I won't have time to share with you today, but it really has driven us to try to understand the kinetics of scale formation in situ in the presence of a membrane surface. So just to sort of summarize from that last section, our empirical kinetic model enables prediction of scale formation under batch homogeneous conditions fairly well for the sulfate system, in particular for the gypsum system. But a key challenge to monitoring kinetics or for predicting kinetics remains heterogeneous nucleation. That is nucleation where the surface, a surface enhances the rate of particle formation. So I'm going to show you just a little bit about a complicated experiment that we worked on to get at this particular problem of heterogeneous nucleation. Led by Chris Takas at SLAC, we used grazing incidence SACs. This method is SACs performed in a geometry that allows us to be sensitive to the interface between solution and a membrane surface. Alex Dachenko, also at SLAC, designed a reactor that allowed us to generate a concentration gradient towards a membrane surface to enable precipitation. And then finally, Jeff McCutcheon's group at University of Connecticut printed polyamide membranes onto porous silicon wafers for us, so that to provide substrates that worked well for this experiment, or this for the GSACs geometry. Okay, so this is the cell that Alex designed. There's a lot going on this slide. What I just want to say about it is he designed a system where we can generate a concentration gradient by flowing nitrogen gas over the surface of a polyamide membrane. A well of solution below that polyamide membrane then starts to evaporate, which drives a concentration gradient from the bottom of the well towards the membrane surface. We can quantitatively monitor that concentration gradient by doing a mass balance on the solution in the cell. And so this is just some trial data that Alex used to show that you get a decrease in, you change the flux of water through the membrane when you turn nitrogen on and when you turn nitrogen off. Okay, so then we take our cell, we put it in the beamline. This is a picture of it actually sitting in the hutch, and sort of in the center, you can see the silicon wafer that has this very thin layer of printed polyamide on top of it. And then we have a solution inlet and a solution outlet that are being connected to a scale, and that scale is connected to a computer so we can monitor the mass of water. And then we have a little gas outlet on top to flow nitrogen over the surface. And when we do this, we then collect grazing incident SACs. And if I tap next, you'll see the world's most maybe not exciting video. And this is actually sped up by 1,000-fold, so it's not real time. But what we see actually is we start to see, we're looking down on the membrane surface, and we can see solution coming through and precipitating at the membrane surface. And this causes a change in the grazing incident SACs pattern. Again, we can quantify the invariant, and that tells us that's a proxy of the amount of particles that are formed in solution. And so in this way, we can match particles being formed to the concentration gradient. This work is new and ongoing, so we don't have a final solution for it yet, but we're really excited because it was pretty complicated to put together, and hopefully yielding some nice results. Okay, so that's really what I wanted to say on the kinetics portion of this. For the last sort of 10 to 15 minutes of this talk, I want to completely switch gears and start talking about the characterization work that we've been doing on silica systems. So before I get deep into this, I want to give you a little bit of motivation and background for why this work is important. So as part of the kinetics goals of this work, we've also been monitoring the induction time for silica as a function of saturation index. We do this using a light scattering method instead of an X-ray scattering method. And we end up with plots of the induction time versus the saturation index. And you can define an empirical or predict an empirical relationship between these two. And the empirical interpretation is a lot like the interpretation of the sulfate system, but underneath, silica formation is completely different from sulfate formation and from the formation of a lot of other minerals. And that's because silica forms via polymerization. So this has been known for a long time. This little graphic I have on the left-hand side of the slide is from 1979. And the idea is that you have silicic acid monomers in solution, that's SiOH4. And those dimerize, and those dimers can then grow into oligomers and eventually condense into particles, and then the particles form via coalescence or aggregation. So when we use dynamic light scattering to monitor this process, we're completely skipping over all of that polymerization. We're just looking at when you have, something of a size that's big enough to be a particle. But when you first start detecting those, the concentration of the monomer has already decreased quite a lot in solution. So on the right-hand plot here, the black line is showing you the decrease in the monomer concentration in solution as a function of time. And you can see that it's already gone down quite a bit before we start to see particles using DLS. So this leads to the question, what impacts that rate of polymerization, right? The rate of particle formation is going to be determined by how that polymerization mechanism happens. And from previous work that I had done looking at scaling on membrane surfaces that were harvested from this pilot scale plant at Orange County, where we had the pilot scale plant had been operated with real feedwaters. What we could see is that there was a lot of silica that was present on these membranes. And that silica was always co-located with calcium. There was always calcium present when silica was present. And we characterized the distribution of silicon to calcium ratios across all particles in the membrane, and they varied across a sort of specific range. And so this led to the question, okay, is calcium just there because it's there? Because it's, there's a lot of calcium. There's more calcium in wastewater effluent than there is silicon in wastewater effluent. Or is it there because it helps those particles form? And so, the first important thing that calcium can do, and also magnesium, is it can help silica particles after they have formed aggregate together, right? And so this is a plot, or this is from previous data that was published showing the lag time, which is basically an induction time for silica in the presence of sodium, magnesium, and calcium. And once you add ionic strength into these systems, the induction time goes down because your particles are coalescing and aggregating faster. So that's one mechanism, aggregation. But in addition to that, previous researchers have already suggested that maybe calcium or magnesium, since they're as divalent cations, can bridge silicic acid monomers or oligomers and that can bring them into proximity, enhancing the rates of polymerization. So we wanted to test this for calcium and magnesium using kinetic experiments where we track the disappearance of the silicic acid monomer over time in solution, and then look for whether calcium is incorporated into the phase or into the phase that's formed. We've done experiments for calcium magnesium. I'm only going to show you calcium today. So basically the way we do these kinetic experiments, we vary the silicon to calcium ratio, keeping the silicon concentration the same, and also keeping the ionic strength the same. So that's important. The ionic strength is always the same, but the concentration of the divalent cation changes. And what we see is if we compare the disappearance of silicic acid without any calcium present, shown by the open circles, to conditions where calcium is present, shown by the red circles, the calcium concentration, or sorry, the silicic acid concentration decreases more rapidly, and it decreases to concentrations that are lower than those predicted in equilibrium with silica when you have calcium present. So the left-hand plot shows just silicic acid monomer versus time. The right-hand plot actually shows 1 over the silicic acid monomer concentration versus time, and that makes it easier to see this rate dependence between 50 on the calcium concentration. I'll note also that getting linear plots in this way indicates a second order mechanism relative to with respect to silicic acid. Okay, so we did those experiments over a range of different calcium concentrations, as I mentioned, and at different pH conditions. And then we tried to reproduce these using a thermodynamic model developed by OLI. And we found that across all of our conditions, we're able to predict the equilibrium concentrations of calcium and silicon in solution using a combination of the phases silica and tobermorite. Tobermorite is a hydrated calcium silicate phase. However, when we do XRD, we see mostly amorphous material. There are some peaks in here. Those peaks are associated with some contamination from calcium carbonates and sodium chloride. There are no peaks associated with tobermorite, and silica, of course, is amorphous. So we're actually more sensitive to these silica and calcium silicate phases using FTIR. So across all a broad range of our conditions, different calcium concentrations, pH 8 to pH 10, the FTIR reveals, of course, the presence of silica through that silica oxygen vibration, but also to the right-hand side of that peak, there's a vibration that's associated with silica oxygen calcium bonding. And so that is characteristic of calcium interacting with silicon. That peak becomes more pronounced at pH 10. So we wanted to take a closer look at these phases. We did that using electron microscopy. So this is, I'll just quickly go over the pH 10 data. We saw some different morphologies of particles, and for the sort of sheet-like structures, we were able to find some crystalline materials, of course, associated with contamination from sodium chloride and calcium carbonate, but we also consistently found lattice spacing at 1.7 angstroms, which we weren't able to identify. So we sort of, right now, we're hypothesizing that this phase is tobermorite-like, but doesn't achieve the long-range order, and its short-range order is distorted such that it's the main lattice spacing of 1.7 angstroms is not actually what you predict for tobermorite. At pH 8, we weren't able to find any lattice fringes at all. The whole thing was completely amorphous. We do, using EDX, observe calcium and silicon to be present. Calcium is present in pretty low concentrations. So this information combined with the FTIR and some X-ray absorption spectroscopy data that I'm not presenting leads us to hypothesize that calcium is absorbed into silica once you get to lower pH. So to summarize our findings for the silica for this phase characterization part of the work, we see that calcium 2+ enhances silica polymerization and is sorbed by silica at low pH. At high pH, it also enhances polymerization, but it may form a discrete calcium silicate phase. We found something similar for magnesium. So the biggest take-home message that I want you to have from this section is that real feedwaters contain multivalent cations always, in addition to silicon. And so the rate and extent of silica formation is very likely going to deviate from that of pure silica. And I think our work demonstrates that both the stability of silica is not adequate for predicting whether silica containing solids will form, and the rates are going to be faster in the presence of cations. So let me just wrap up and reiterate that a predictive understanding of fouling and scaling is going to require a combination of thermodynamic and kinetic modeling along with mass transport models. And we really need, moving forward, foundational understanding of what materials are present and their stabilities in order to develop thermodynamic models. And then a step further is developing those kinetic models for those systems. So we're able to do the kinetic modeling for fairly simple systems, but the crystalline sulfate minerals are one such system. But these kinetic models present a major challenge due to the importance both of heterogeneous nucleation, and I didn't get into this much, but also due to the presence of non-classical both nucleation pathways and growth pathways. And so it'll be really interesting moving forward to see how those mechanisms of growth are can be described and are impacted by mass transport in the system. So with that, I'd like to thank everyone for coming and attending this webinar today. And I also particularly want to thank a couple people who are part of the main NAWI project that I have funded, but who are really instrumental in doing the work. Chris Takas and Alex Dadchenko for the heterogeneous nucleation experiments, and then Pete Dahlberg and Jacob Summers, who worked with Sharon Bone to perform the cryo-electron microscopy experiments that have been really just great for gaining insight into the system. So with that, I'd be happy to take any questions.
Yeah, thanks, Sharon. Really appreciate the presentation. I was fascinated by a lot of that and the methods that you're going to determining everything from nucleation to what is actually forming on those and so forth. So we have a couple of questions here. One came from Leon Auerbach, who's asked, did you look at the impact on solubility of calcium sulfate with magnesium and temperatures which can have reverse solubility and also can have a stable form of hemihydrate with high temperatures? He says that's often the case with thermal desal and crystallization. Yeah, I mean, the short answer is no. And we didn't in particular because we have to cover a very broad range of phase space for these experiments. And I think that goes specifically to the point that these one has to think pretty carefully about the composition of solutions that are relevant in a different setting. And there's a lot of work to be done to develop, in particular, kinetic models for mixed systems. We don't have kinetic models that are sufficient to look at
to do modeling for even like the standard minerals like the sulfates and the carbonates, and then when you add in other materials, that just expands your matrix hugely. So there's a lot of work left to be done.
Yeah, I would agree, and it makes me think of some things I observed last week at visiting some different plants, which I'm going to ask in a minute. But I'm going to get to Peter's comment and questions that he had here. And Peter, I don't know if you want to come on and ask those since you can.
So Sharon, great talk, and again, we're diving into an extremely complex geochemical system. And I guess two questions. One is just observationally, calcium and magnesium love silica. And in very low pressure systems like stellar systems, that you form calcium magnesium silicates very early in condensation reactions. In molten geochemical reactions, calcium and magnesium love silica, and they form some of the highest temperature phases, et cetera. So in a way, not surprising that you add calcium and magnesium and old friends get together and facilitate some sort of bonding. The second thing is, I was wondering, given the IR work, if you guys had explored NMR to look at next nearest neighbors and look at incipient crystallization or nanocrystals, et cetera.
Yeah, no, that's a interesting thought. We hadn't tried NMR. I mean, mostly due to a lack of a team member who's
you need an NMR nerd
experienced.
somewhere.
Yeah,
right?
yeah, but I think like for the nanocrystalline, the potential nanocrystalline character, I think the TEM is actually quite good. For sort of understanding nearest neighbor contribution, or what the structure is around the calcium, that I mean, I'm sure NMR would be really great. We do have X-ray absorption spectroscopy data, and that also provides some information on coordination environment.
But average. I mean, and the interesting thing about NMR, you also can get dynamics because of the exchange rate, and that's kind
okay.
of,
Yeah.
Last thing is, you're looking at, welcome to geochemistry, Sharon. The periodic table, it's water, it's a mess. The practical thing for operations is, how far can I drive a system in steady state before something's going to show up? Or how can I cleverly tick-tock between squeezing water out of the system and then backing off and back flushing? And so I guess it's not really a question, but one of the areas people use antiscalants to suppress the sort of steady state nucleation phenomenon, and then they use non-steady state osmotic processes to form nanocrystals and then wash them or kind of redissolve them with a backwash step. So there are sometimes operational ways in which you can, I would say address, but try to fight against some of the inevitability of nanophase formation. And this is all to say, more a comment to everybody in the community. This issue of scaling remains one of the enduring challenges for optimizing reverse osmosis processes. And so there's no end of work to be done. But I mean, I'm really excited by how you've lifted the lid on some of this, Sharon. I want it to continue. So what would you guys like to do next in terms of a set of experiments or a set of
Yeah, good question. I mean, so there's a couple different areas that I'm really interested in, and one is developing a fundamental understanding of the interaction between scalants and the membrane surface. And I think there's a lot of work to be done to expand the model system for the heterogeneous nucleation to enable us to understand the drivers of heterogeneous nucleation for different for different scales. In particular, contrasting something like silica versus a strontium or a barium sulfate.
Yeah.
And I mean, I think also beyond that, I'm particularly interested by silica. I'm particularly interested in it because I think that's a really problematic one, right? Like
I
when it
totally agree.
forms, how do you get it off? And so I'm really curious about whether you can decrease remove other constituents that might enhance silica formation and help prevent silica.
You
or
can fight silica directly, or you can take away all its friends. And
Yeah,
try to
you
get
can.
rid of its friends, and so it's then
Yeah.
the remaining propensity is a little bit
Yeah.
less. Yeah, it's an interesting point.
Yeah, and I think I think it's an important point that let me put it this way. On the Orange County membranes, there's silica everywhere. It's everywhere. And the average concentration of silicon in the incoming water at Orange County is 20 milligrams per liter.
Yeah.
That's 5 times under silica
Saturation,
supersaturation.
quote unquote saturation. Yeah.
Yeah. And so then even if you have concentration polarization of concentration polarization factor of say 4, presumably you're not oversaturated with respect to silica. So why is there so much silica there?
Yeah, why is it on the spacers, and why is it on the gasket? Why is it on a bunch of stuff that's not got a flux associated with it? Yeah.
Yeah. And so I think like it's probably a combination of needing better mass transport models to understand why, like what the fluid is doing around those spacers and why materials accumulating there. And then also probably needing to better understand the stability of the silica material that's forming, because it's not pure silica. That's sort of the hypothesis that I feel like this work and that prior Orange County work.
It's nanophase olivine. It's nanophase, some of these weird hydrated calcium, magnesium, silica.
Yeah, I think a lot of it is like amorphous hydrated silicates.
Yeah.
Yeah. And then the last thing I'll say is I think this point that you have phases where you have substitution, like the previous question alluded to. I think there's a number of those phases that are also relevant for scaling and be interesting to pursue.
Yeah. All right. Well, thank you again.
Yeah, thanks, Sharon. We do have a couple questions left. Let's try to get them in. So from Thomas Septon, he says, great work, thanks. And he says, at what point do the calcium sulfate rod aggregate into larger particles? In the cases of a system where you are seeding calcium sulfate particles to compete with scale formation on surfaces, what is a good seed particle size to get rod formation and suspended aggregates?
Oh, I'm not sure if I know the answer to that, like what a specific size would be, but I think that I think those rods form extremely rapidly. And they form rapidly. I think that most of the time that gypsum spends forming is growth of those rods. I think so if we think about the time from mixing of calcium with sulfate to the time at which gypsum reaches equilibrium, the vast majority of that time, say 95% of that time, is the rod formation, or is not the rod formation, is the rod growth.
Prenucleation, yeah.
And the 5% goes from prenucleation the 5% of that time is devoted to prenucleation clusters to the onset of those, or the first formation of those large rods.
Right.
That's not, don't quote me on that time scale. But I think if you look at the comparison of the how long it takes the calcium concentration to reach equilibrium versus when we first start seeing those rod particles, I think that most of your time is spent on rod growth anyways. And
Yeah.
yeah.
So another question came from Kofi Christie, who's asking, he says, in the lab, we notice much variability in scaling induction time. Is the major cause more a consequence of thermodynamic metastability or perhaps user error?
Oh, good question. I think it depends on the system. It depends on how you're performing your experiments. I think, and I like, yeah, it's quite possible it could be user error. We do have, right, we do replicate experiments, and that does add error bars to our calculations. And I think, I think a lot of it is actually just like the small deviations in concentration add to the error bar. And then I do think you have to be very careful, particularly less so for calcium sulfate, particularly for strontium and barium. They're so sticky. You really need to be careful about the making sure the barium and strontium is completely cleaned from the surfaces that you're using. I think that actually has been probably induced some error in our systems. Yeah, and then, but I think for the metastability, yeah, I'm not sure. I mean, it's a good idea to characterize the final phase that you're forming, but also I think it's important to recognize that when you form minerals in a not very controlled environment,
right,
you might be going through a metastable phase. And so there's actually evidence not from us, but out there in the literature that the way we're forming gypsum, you could go through a basanite phase first. I don't know, like there's some evidence for it. It's not for sure, but yes, it's worth considering. Is it going to introduce error into the induction times that you actually measure? I think probably not, unless you're not controlling your system such that your end product is you're not making what you think you're making.
Yeah, right. Very interesting. Well, Sharon, I think with that, we're going to bring it to a close. We really appreciate your presentation and we appreciate the audience. And we look forward to the next NAWI webinar. So thanks so much, Sharon. Appreciate your what you presented today.
Yeah, thank you so much for having me. Thanks, everyone.
All right. Thanks, everyone. See you next time.